Thank you. So we can continue. The next speaker is David Copplestone from University of Stirling in UK, and he's going to speak about the protection of the environment, which is something that ICRP is looking at it very seriously, and he's trying to respond to the question, where is the radiation protection world today and what lies ahead? The floor is yours.
Okay, good, microphone's on. Thank you, Jack. Good morning, everyone. So I'd like to talk to you today about some of the issues related to protection of the environment. And given that it's a relatively new field in radiation protection, perhaps being developed over the last 20 years in detail, I would say that looking forward, we are still facing a number of problems and issues that we need to address. So my plan is to try and look at some of the drivers behind why we need to be thinking about protection of the environment. And then I'm going to use the opportunity to give you some information about where we are at the moment, and then using some examples, try to illustrate some of the issues that perhaps are more important or need to be considered to help to address the issues over protection of the environment from a radiological perspective. So if we have a look at some of the drivers, first of all, the need to move to a low-carbon economy, I think, is fairly self-evident. Accepting the comments earlier made before coffee, low carbon could be dealt with in some instances by a new generation of nuclear power stations. The point here is not about getting into a debate about whether that's a right or wrong thing to do. The point here is that if we're thinking about protection of the environment, we need to be prepared for understanding what will happen if we suddenly have a new tranche of reactors coming on stream and so on. How do we deal with the radioactive wastes that are arising from those? Because many countries are struggling with that as an issue in terms of the final disposal solution. But interestingly as well, whether you agree with nuclear power or not, I think most of us in the room would probably want to be able to make use of nuclear medicine, radioactive isotopes and so on for treatment, for cancer, etc. And so what we're finding is that there are increasingly new radionuclides coming out into the mix that are being used for new medical treatments and so on. Now, I'm no expert on this, so don't question me on which ones, but that's certainly a factor in this consideration. And something else as well that's coming in, certainly in the UK, is there is a tendency to move large cancer centres together and amalgamate them, which it means by default potentially larger releases of radioactivity than we've seen in the past. So again, does that have any consequences for protection of the environment? A lot of this has come about through the interest in terms of conservation, and so that has also been a driver, certainly in the last 15 years or so, thinking about how we can demonstrate that the environment is protected from releases of radioactive materials from different operations. And I think one of the issues here, we've heard mention already of things like public interest, public safety and so on, but there's also a high level of media interest, and I'll come back to that later on, in terms of these issues. So I think we have a number of things that we need to be trying to address into the future when we're looking at protection of the environment. So where exactly are we at the moment in terms of the statements that are being made? Well, if I look at ICRP publications, if you look at the recommendations in publication 103, then it stresses here that the primary aim for the Commission's recommendations is to look at the appropriate level of protection for people and the environment against the detrimental effects of radiation exposure. And if we look a little further into the recommendations, you will also find in the recommendations in 103 that this is about preventing the frequency and number of deleterious effects that might occur, particularly with respect to those conservation drivers, so thinking about things like biological diversity, conservation of species, ecosystem structure and functioning, and so on. In addition to that, if you look at the IEA documentation on this issue, they're also stressing the fact that we need to be thinking about these things in the context of resources that we may also require for ourselves in the future. So there's a number of different protection goals that have been put forward within the context of protection of the environment. And that means we have to be able to think about how to do assessments, we have to think about how we're going to implement within the radiological system of protection the need or the requirements for wildlife dose assessments. And that goes through the term of reference animals and plants that is brought in 103 and expanded on in publication 108. And looking at that in terms of the publication 108, you can see here for our different exposure situations, so planned, emergency, and existing, and again, I'll come back to those later. All of those will lead to radioactive releases into the environment, and those releases will give rise to environmental radionuclide concentrations that we then need to do some assessment with. And depending upon whether we're looking at humans or wildlife, we will start to go through the process of looking at reference person or the reference animals and plants. And I'll come back to the term for the wildlife, the derived consideration reference levels. I'll come back to that later on. But the derived consideration reference levels, or for humans, dose limits, constraints, and reference levels, are there to help us to make decisions. They're there to help us to think about the consequences of those releases of radioactive materials from whatever source that we're talking about, from whatever type of exposure, to think about the consequences to humans or the wildlife, and how do we use that information to make decisions, and how can we help make sure that we are using some of the ethical principles, and I'm going to try and get this right because I usually say it the wrong way around, to do more good than harm when it comes to the environment. Okay? So I want to take a step back for a second. I said we want to be able to do an assessment. Well, I'm going to go back to a very simplified slide in terms of the considerations for how we think about the different components of the assessments. So this is for humans, and this is looking at our radionuclide source, our exposure pathways, and then using things like habits data, and it's in yellow for a reason, as you'll see in a moment. We think about the consequences of the exposure for us as individuals when we're eating more or less than other people, or that we're spending more time in the environment where there may be considerations in terms of exposures to radioactivity that's been present in the environment. So where you might walk your dog, for example, and so on. And all of that can be turned in through the use of the reference person concept. We can turn that into a dose that allows us to then consider that dose against the markers, in this case for humans, the limits, the dose constraints, and so on, to actually be able to consider the consequences of that exposure. Now, I hear lots of things that people say that it's different when we come to doing this for wildlife and so on. Well, the reason I've come to this simple level is because I want to show you that actually at this level, I don't think it is that dissimilar when we're trying to integrate the exposure assessments for wildlife and humans. So hopefully what will change now as I move to the next slide is the little bits in yellow will change because those are the things that are really different. Excuse me. So for wildlife dose assessments, what we're interested in here is understanding the similar things. So we still need to know what our radionuclide source is. Well, one hopes if we're doing this as a combined assessment, that's going to be the same source. We're looking at pathways of exposure. Now, they're going to be different to the wildlife, and they're going to revolve around these, what I've termed here, ecological parameters, where the animals spend their time in the environment and so on, and how they're exposed to the radiation. And we're going to use the reference animals and plants concept here rather than the reference person. But other than that, we're going to calculate the total absorbed dose, and we're going to compare that to something. In this case, we're going to use those derived consideration reference levels that we're going to be looking at. Now, I want to take this a little bit further and break this down into 3 component parts. And I want to look first at how we can do the dose assessment for the wildlife, and then we'll look at the dosimetry, and then we'll look at the effects side, because I want to look and see how we might need to do more work in the future on each of these topics. So if we have a look at transfer, first of all, which is where we're trying to establish the concentration, the radionuclide concentration in the biota of interest, we're usually doing this in a simplified approach. We're usually using something like a concentration ratio to estimate the concentration in the biota over the media of interest that we're working in. So it might be water, air, soil, etc. And what we're trying to do here is capture all of those different component pathways. And we do something similar for humans, again, in terms of the use of factors like concentration ratios and so on. But there are differences. And if we look at those differences for a second or two, that's really related to the fact that the wildlife databases that underpin the assessments are looking at whole body concentrations of these radionuclides. We simplify things down to whole bodies, and we don't necessarily work on different tissues within the bodies. Whereas for humans and so on, obviously, it's about what we eat and what we consume, so we're interested in the concentration ratios to the foodstuffs that we're interested in eating. Other things that come into play here, and that's increasingly becoming part of the questioning over whether we're capturing everything when we're thinking about protection of the environment, is perhaps that we don't necessarily think about all non-human species when we're considering the assessments that we're doing. In many cases, these assessments that we're doing for wildlife are for wildlife. They're not for domestic stock. And so again, there's a question over whether we need to expand or consider how we're connecting to livestock in terms of this process. And some of the issues that we have in terms of dealing with this, especially when it comes to those planned exposure, planned existing and emergency exposure situations. Emergency situations are never going to be in equilibrium in the environment, so they're going to be dynamic, and we haven't necessarily got all of the assessment criteria in there that we need. In addition to that, if you think about the wide range of species that we have to deal with in the environment, and you think about all of the radionuclides that we're trying to assess for, then we often have missing data from the databases that underpin those wildlife dose assessments. And so some of the new things that are underway are different ways of trying to fill in those data gaps. And I mentioned something on here which is called REML work. I'm not going to go into this in any detail, but this is a way of being able to rank different species against each other to try and help us to understand the likely transfer factors that may be present for certain radionuclides that will actually allow us to think about using proxies to fill in missing information. So certainly for transfer, there are some differences between human and wildlife assessments, but in general, we're starting to be able to do these assessments where we have the data. If we move to dosimetry and think about the future for dosimetry and where we are with dosimetry now, for most wildlife assessments, we're looking at simplified geometries. Again, given the wide range of species, the wide range of species and habitats that we're trying to deal with, we're using simplified approaches, spheres, cylinders, ellipsoids, and so on. In some cases, we've done some work that has looked at the presence of radioactivity within the ellipsoids in different positions. Hence, you see in the graphic on here, the small dot on the right-hand side of the slide, and so on, where we can think about the consequences and the uncertainties associated with if the radioactivity is actually not homogeneously distributed within the body. But actually, from a scientific point of view, there are other issues why we might need to know more about the dose-response relationship between particular organs in a body and those of the effects that we're interested in. And this isn't, I stress this before I go on to the next slide, this isn't because we're concerned that the dosimetry isn't adequate. This is about testing the dosimetry to see whether we are using things that are fit for purpose. So some of you probably worked out where I'm going already. And in human radiological protection, there is now a move to using voxel phantoms, and in some cases for scientific interest, and not for regulation purposes, we're interested in looking at the distribution of radionuclides within the body of some of the reference animals and plants. And here's the crab that's been through a CT scanner and looked at in terms of the different distributions. And that's very much at the moment for testing those models. Are the simple approaches that we have fit for purpose? And it's very much about trying to help us to understand the dosimetry when we're looking at dose responses in the effects literature. And I'm not going to go into any more detail than that, but if anybody's interested, we can talk about it afterwards. Because I think one of the biggest areas that we need to work on is actually these dose-response relationships. And the effects information that we have is one of the key areas to make sure that those derived consideration reference levels are actually appropriate and fit for purpose in terms of their potential use for establishing whether something in the environment is likely to have an impact or not. So what are these RAPs that I keep talking about? Well, the reference animals and plants are listed here. You can see them, the deer, the rat, the duck, the pine tree, and so on. And on the other side, on the left-hand side, as you're looking at it, you can see the wildlife group that they're reflective of. And one of the problems we have when we talk about the RAPs is that everybody says, well, where's my species of interest? Well, that's one of the questions that we'll come back to. But if we just take them as they are for now, these are a means for us to be able to construct a system of radiological protection to look at the consequences of dose exposures to these particular types of organisms. And they are trying to help us to derive these derived consideration reference levels. And these are the bands that are on here in terms of milligrays per day for each of those organisms, okay? So what is the derived consideration reference level? Well, it is a band of dose rate that is there, which might indicate where we have some chance of deleterious effects. I say might because in some cases we have very limited amount of data on the radiological impacts. One of the things that also comes out in this is that this is about individuals usually, because they're not always about populations. So that's an area of interest as well, which I'm not going to talk about any further. But this understanding of how we're protective of a population of a particular species is something that we also need to consider over the next few years. But when we come to apply these derived consideration reference levels, we can go back to thinking about planned, existing, and emergency exposure situations. And on the left-hand side of this graphic, you can see the use of the DCRL with respect to planned exposure situations. Now, when we plan to release something into the environment, it would make sense, it makes sense to me, and hopefully conceptually it makes sense to you as well, that we wouldn't want to be causing harm to the organisms in the environment. And therefore, the idea would be that you wouldn't want to be going into the DCRL band. Remember the definition? It's where there is some chance of deleterious effects perhaps occurring. And at this point, I usually say that that's my interest in planned out of the way, because I think they're pretty boring from a radiological protection point of view of the environment. Because we shouldn't be causing effects. We shouldn't be having those concerns. And we should be dealing with this from a human radiological point of view. That means that the discharges are low enough that it's unlikely to have an impact on the environment for the wildlife that we're interested in. What's potentially more interesting are where we're looking at existing exposure situations and emergency exposure situations. And how do we apply and think about protection of the environment in those contexts? And the assessments allow us to work out what the dose rates are to different wildlife species and so on, and to think about the consequences of their exposures. And it allows us to then know where we are in relation to the DCRL band. And for existing exposure situations, it would make sense that we wanted to reduce the harm in the environment, and therefore we might want to work towards reducing to or below the DCRL band in the environment. Now, optimization of the protection for both humans and wildlife in that context. But I think we can learn a lot here from what's happened in the past. And I think one of the things that we need to think about for the future is actually providing much more advice and guidance on how we actually think about that implementation, what's good and bad practice, what could actually cause more harm to the environment when we're considering the consequences of actions, either for the purposes of reducing the doses to humans and or for considering for wildlife in that situation. And similarly, when it comes to accidental scenarios, we're not really going to be thinking about the time where we're interested in the concept of running in and picking up all the animals and running out from the contamination that's occurring. But what we can do with the DCRLs is fairly quickly start to think about what kind of effects might we see in the environment should there be an accident, which hopefully there wouldn't be in the first instance. But even here, it's perhaps thinking about protection of the environment and embedding that thinking about, are there ways that we can build something that would actually minimize the impact on the environment? Can we site it in a different location that might minimize the environmental impacts and so on, especially if we're interested in, say, conservation, and there are certain areas that we want to avoid in the wider environment for our protection purposes. So these DCRLs are starting to allow us to think about these concepts within the three different exposure situations that I've mentioned. What then are we worried about for the future? What are the challenges that might be coming that we know about already? Well, I want to talk about and give you some examples of challenges coming in the literature from fieldwork, which is looking at the impacts of radiation in the environment. Excuse me. And I want to talk about the potential consequences of those studies that are coming out, that are published, that are in the literature, that we need to be thinking about and dealing with. And that leads to a question about whether the DCRLs are actually in the right place. Do we need to think about whether the numbers are in the right place and so on? And also, how do these reference animals and plants represent that wider group of wildlife organisms that I mentioned earlier? In addition to that, I think one of the key things that seems to be coming out of a lot of the effect studies at the moment on wildlife is that we're seeing what I would class as a subtle effect of radiation exposure. We're seeing changes that aren't necessarily going to cause things like reproductive effects in that species, but maybe are going to start to have combined effects that we need to be thinking about, and I'll come back to that later on. And then we have the old problem and issue of dealing with radiation as one stressor of many in the environment. But if I look at the field studies information for a moment, I'll just give you a couple of examples from news stories that we've seen appearing in the press. And this is just a couple of studies that one says Chernobyl species decline is linked to DNA, and on the other one it says wildlife defies Chernobyl radiation. So this is immediately suggesting that we have two very conflicting sets of information going on within the literature. These are both based on scientific studies that are in the published literature, but have very different findings. And I want to explore what the consequences of that are, because there are some significant statements that are made in some papers about the consequences of the radiation exposure. I'm going to look first at some mammal data to highlight the issues here again. So here you can see a relationship based on this published paper that has suggested that the radiation levels are causing a reduction in the abundance, in this case of 12 species of mammals, with a very high degree of probability in this context. Now, if you look at that pattern of distribution of data, and I should say at this point, we don't know how many data points underpin each of those open circles. There may be more than one data point under each of those circles. But I look at that, and if you exclude the 3 sites here across the 100 microsieverts level, then to me, that's suggesting that there isn't really a relationship. It's only when you put those influential points in that it's actually driving a relationship, and then you can even question the significance of that. But, and this is the point I want to make, before we go any further, these are the data that are challenging the circumstances about radiation protection of the environment. These we know that regulators are being challenged with in terms of here you've got evidence that mammals are being affected by radiation exposure at what I would consider to be relatively low levels of radiation exposure in the exclusion zone. So just to try and unpack that a little bit more, we've done some further work on this particular paper to try and understand it a little better. Those, as it seems on the graph, 3 points that are circled there, those are actually, we think, coming from the red forest, because that's the only place you see exposure levels of that level in the environment. And so if it's the red forest, we know that there's anthropogenic activity in that area and nearby. We know also that the red forest suffered significant damage and isn't the same ecosystem as you'll find elsewhere in the exclusion zone. So there may be other reasons why mammals aren't present in that area anyway. So that's one of the things that we need to understand. However, from a radiation protection point of view, these data are out there and we need to understand the consequences, especially when we add in the co-study or another study that in that green box has shown no relationship. So I haven't put the data here, but I'm just showing you that over that range, in another study that's been published, there is no relationship to decline in radiation, which is, I think, what most of us would have probably interpreted if you'd have seen that graph without me putting a line in it. We have another study. This is another study looking at the impacts, looking at soil leaf litter decomposition rates. So this is the breakdown of litter in these litter bags that you can see in the photograph. And on the left-hand side, you can see that there's a reported decline in the activity or the rate of decomposition that's going on with increasing radiation. And on the right-hand side, you can see another study published from the exclusion zone, which suggests that there is a different relationship. In this case, that as the radiation levels have increased, there's actually an increased rate of decomposition. Again, from a scientific point of view, how do we handle this type of information? How do we deal with this in terms of the impacts on wildlife? And last study I want to show you in this context is this one here, looking at invertebrates. These are the different types of organisms, so butterflies, bees, and so on. I don't want you to understand the units at this point, but what I want you to note is that in each case, you can see a very negative association with the radiation increasing on the x-axis against the abundance. So this is the presence or absence of those species at dose rates across the exclusion zone. I expand that up now for a second because I want to stress this. If you overlay this graph with the numbers that we think are protective at the moment, then again we have an issue because if you look at, for those of you who know about the European project Erica, that was our predicted no effect level that we said, and look at that, below that graph, all the grasshoppers, all the bumblebees, all the butterflies, and all the dragonflies disappear. But that's our predicted no effect level. If you look at the ICRP, DCRLs, again, most of the effects that are being reported in the literature are suggested to be below those numbers. The thing that I don't get, as those of you have already probably noticed, if you look at the final number that I put onto that, you can see the UK natural background as a comparator, and we find across the UK different areas that will have that kind of level of UK natural background. And I know there's lots of people out there who will say that butterflies and so on are declining in the UK, etc., but I am pretty sure it's not related to a sudden change in radiation levels. I think that might be something to do with other activities that we have done to the environment, not related to radiation. Well, okay, isn't the simple answer that the numbers are in the wrong place? Aren't the DCRLs in the wrong place? Aren't they, the safety factor that we'd estimated for Erica, isn't that simply in the wrong place? And indeed, that could be the case. And if we look at a particular example of some recent data that we have from the radiation effects studies that we've been doing, you can see here that this is for the reference B. You can actually see in the range of sort of natural background up to about 10 milligrays per day, we didn't have any information in the DCRL bands. So we set out to do an experiment to do this. And this experiment was actually done by one of my PhD students. And Kat has exposed bee boxes along a radiation gradient to look at the consequences of radiation exposure. You'll see on the x-axis that we go up to about 72 milligrays per day. The bottom of the DCRL bands was about 100 milligrays per day, so we were trying to test the DCRL bands. We were also looking at dose ranges that cover those that we see in Chernobyl. And we did find some interesting effects, and it would cause us pause for thought in terms of thinking about where the DCRL is for the reference B. And these are the data, and you may well look at these data and go, blimey, it's just the same graphic again that we just talked about, and there's influential points and so on here, etc. Take your point on that, and I can go through that with you in detail later on. All I will say to you is that Kat has taken away these two extreme points and seen whether there's still the relationship. We've taken away the top ones on the y-axis and so on. They've tested the statistics in various different ways, and we are confident that the relationship still stands. And at the 75 milligrays per day or thereabouts dose rate that she was giving rise, there were 84% reduction in queen production. And queens are a significant investment in a colony of bees. They are the reproductive important component within the colony. And even at dose rates below that, we still saw reductions So that 28% is at the bottom of the next DCRL band down. And even that, we think, is likely to cause problems in the environment if you've got that kind of level of reduction in queen number. And we get that from work that's been done on pesticides. So there may be some evidence coming in. I will stress there were no data before. There are now some data that will be published very soon that might influence where we position the DCRLs. They don't drive the DCRLs down to address and understand the data that I just reported to you about mammals and so on from the field, however. Not by a long shot. And just to show you a couple of graphics, the orange ones on here, I'm not going to go through this in any great detail, but this is just some work that's been done collectively within the tree project looking to test the DCRLs. Those in orange have some level of effect. So you can see them relative to the black bars, which are the current DCRL bands. So the main one that I would highlight here is the invertebrate one, which we think is probably in the wrong place, based on the studies that have been done. And if you look at that in field-based studies as well, you can see those kind of consequences coming in as well here, with possible addition of a couple, and I mean a couple, of fish that demonstrated some effects on the fish in Chernobyl. Not sufficient numbers to cause me to be concerned that the DCRL is in the wrong place at this point, but indications that things are happening at lower doses than we might have expected based on previous literature. And this is important because those DCRLs are going to be used, and what we need to also understand is how those DCRLs are in relation to other bird species or other species and so on. So forgive the graph on this. This was hand-drawn yesterday just to illustrate this point. But the question here is, where does the reference duck sit against all bird species? Because that really will help us to address the questions that we keep getting asked about how does my particular species reflect within the DCRL bands and so on. And so all of this is really starting to think us about the consequences of exposure. How am I doing for time, Jack? Am I... Not long, I don't think. Okay, okay. So I want to just look at another piece of work that's been going on, and this one isn't going to show very well on this graphic. I apologize in advance. But what we've been doing is we've been looking at the impacts of radiation on different species, and one of the species we've been looking at is the Daphnia. And what we've done is we collected Daphnia from different lakes within the exclusion zone, brought them back to the laboratory, and we've then carried out some phenotypic measurements of fitness on those particular individuals that we brought back. What this graphic shows, believe it or not, and you might have to trust me on this one, is that as the radiation levels in the lakes increase, we don't see a relationship, for example, on the number of offspring that are being produced over the experimental day period. It's not a relationship with radiation exposure. I should say that these aren't exposed in the lab. This is what's happened to them in the past. But, and I say you might have to trust me on this one for this graphic at this point, what we do see is a shift in the timings. And so here, for example, you can start to see that there's, I don't know if this will work. No, I can't see it. You can see that the graphic, the hump of the graphic is slightly in different positions relative to the lakes. Oops, and I've gone backwards, and I haven't gone forwards. And that is perhaps something that is happening in terms of the relationships that are happening here. And I'll come back to that because I think this is a really interesting effect that we're seeing. These are timing differences. They're not going to cause problems in terms of what we would perceive as the structure and functioning of that ecosystem. So what does this all mean for the future, and what do we need to be thinking about and the consequences of this? And I think there are a number of scientific questions that come up. One of the key things that we need to understand still, for the broad range of species that we're interested in, is what is causing their vulnerability to particular radiation? Why are some species more or less radiosensitive to the radiation that we're interested in? And how can we demonstrate that the DCRLs are protective of those wider range of species? I've just mentioned that one about the subtle effects of radiation. Now, I did that with Daphnia, and Daphnia perhaps don't really matter in that context, but now let's think about this from something like plants. If we're starting to see a change and a shift in plants, and we do have some data for this that I haven't presented, that the timings for plant flowering is shifting, now put yourself into the field and put your mindset as being a bee. You've come out of hibernation and you're looking for some food. That food isn't present. You could have some consequences in the environment that we weren't expecting. So this delayed production of pollinators, we could have delayed problems with pollination services as a result of this. And all of these types of questions are starting to make us wonder whether there are bigger problems that we think for what are relatively high radiation levels still in the exclusion zone. They're still not anywhere near what we would see around normal planned releases. So remember, this is for existing exposure situations, okay? But we're starting to see that kind of information coming out in the literature. Last one. What are the relationships between radiation and other stressors? And again, this isn't something that keeps on coming up, and that is actually a problem because here ICRP has a remit for radiological protection and doesn't have a remit for non-radiological contaminants. So just to summarize all of that, I want to say that there is an increasing need for demonstration of the protection of the environment. We need knowledge on the effects to help us to regulate the uses of radioactivity, whatever those purposes may be. And we know that people are challenging the understanding of the radiological system for radiation protection of the environment from the basis of these field studies. And I saw some of you smiling and so on about the data I was presenting, and we probably have similar issues in that sense. But these data are published. That does suggest that something is happening. Whether it's radiation and other stressors and so on is something we need to understand and establish, especially if we want, in 10 years' time, a fit-for-purpose, robust and fully integrated system of radiological protection. Thanks, Jack.
Thank you. Thank you, David. Right on time. Perfect.
Okay.
Well, questions? Ted.
Thank you very much, David. That was, as usual, very interesting. One of the things that you pointed out, you focused on the DCRLs, focus on, in essence, individuals within a small population. And for environmental protection, I think that's completely appropriate. We're not worrying about the individual. However, we probably are worrying about the population and maybe about the ecosystem. So how does that fit into the future plans of what's going on in terms of addressing those sort of health issues?
Yeah. So hopefully the comments I just made about these subtle changes may well lead to more understanding about the ecological ecosystem level type impacts. But that's the case at the moment, that's come out of the data that's been done in the field and the laboratory in the last few years. I think we need some, unfortunately, some support and some funding to help us take that further forward. In terms of the individuals and the populations, there are other ways we can look at some of that, and there are some activities going on with population modelling to try and explore some of that. That's not going to be for regulatory purposes before anybody starts to panic, but I think it again is about testing and looking and seeing what the consequences of changes in the individuals might lead to at a population level. So.
Okay, first, there.
Synnerve Bergman, Vattenfall AB. Could you explore the second point there, when you say need knowledge on effects of radiation? I guess you mean on the environment to regulate effectively nuclear power. What do you mean with more knowledge here? Is it the regulation? Is it nuclear power, or is it on the effects?
It's the effects. Sorry, the bit in brackets is my. This is how it will be used potentially, okay? It's not meaning that we need more data just for nuclear power in its own right and so on. The problem is, as I've just explained, hopefully, we have these conflicting studies in the exclusion zone, for example, and the understanding that's coming from that is suggesting that radiation effects should be occurring at lower doses if you believe those data. And those data are not in isolation. We're seeing similar reported information in the literature from the Fukushima, around Fukushima and so on now as well. And the problem is not that they're showing data that are effects at what is an existing exposure site. I get that. Some of the dose rates that they're reporting at are below what I would consider to be numbers that we would use in the environment. That's where the issue comes, if it's proven to be true. I'm not standing here and saying that. If you want my honest opinion, I would say that there's something not quite right with the way that those studies have been done, the way that the information has been interpreted. Something is not quite right. We're trying to unpick that. And the need for more knowledge is to address that.
Pierre? Don?
Thank you very much, David. And it's more as a comment, and then I think echoing what you are saying. I think it's very important what you indicate, that we have to look beyond or look at the whole ecosystem. For example, in what has been done in UNSCARE, following up the different accident and through consensus seminars and conferences, it is prioritized this with going from laboratory to the ecosystem, look at the combined effects and how one effect may occur in the ecosystem, which is not that part of the ecosystem which has been exposed. So I think if one are thinking of in the future what is coming out about the effects, for example, from UNSCARE and IOR, will lead to input to how should you manage this in a quite a different way than we are doing at the moment. So thank you for that.
Okay, thank you.
Last question to Don.
Thank you, Donald Kuhl from the United States. Actually, a comment and then a bit of a question. The first, the comment, just so that everyone can be aware, that you had mentioned at the beginning that an area that is yet to be looked at is the question for livestock and other animals. In fact, ICRP and the main commission this week has started a process to start to look at situations in which there is a more one-on-one husbandry relationship in veterinary practices, in zoos, in livestock, and a variety of things to come. The question that I wanted to ask you was whether there was anything that you could elaborate on with respect to radiological challenges and other environmental challenges together. As we had talked about with radon just before the break, I think we have to be careful not to look in isolation of just radioactivity, because there are many bits that may be contributing, and it wouldn't necessarily be surprising that some of them might even be more than simply additive.
I probably haven't got very long to respond on that. We can pick that up afterwards.
Sure.
But that is exactly the kind of thing I was talking about that need knowledge on the understanding of where these studies are coming from, why these studies are there, and what they're finding may not be related to radiation at all. It might be simply to do with human activities and so on. It could be to do with past exposures. It could be all sorts of potential reasons why we're seeing things that could be happening now in the likes of the exclusion zone around Chernobyl and Fukushima from these literature studies. Lots of us want to dismiss these studies, but they are there and we need to be able to address them, not to dismiss them. So.
Thank you. I'm going to close the discussion now. We can continue during the panel. Thank you once again, David, for your very clear presentation.